Welcome, everyone. I think we're going to go ahead and get started here. It's 4. It's great to see so many familiar faces here, faculty, staff, students, and postdocs. This is the Living Earth Collaborative Thursday seminar, and this is the second of 2 seminar series where we've had our resident graduate students here at Washington University in the Evolution, Ecology, and Population Biology program come and share some of their research that they're conducting for their dissertations. So first, we have Lauren Johnson presenting today. She did her undergraduate at Trinity University and graduated with a BS in biology in 2021. Lauren is a second-year student in the Wassel lab. She is presenting today on the hormones and behavior in anolis lizards. Lauren, take it away.
Great, thank you, Ari, for that introduction. For the day's talk, I'm actually going to roll the tape back just a little bit and present on some work that I started as an undergraduate at Trinity University in Dr. Michelle Johnson's lab. And as some of you know, Michelle Johnson was also a former EEPB grad student at WashU, so it seems really fitting for this talk that I'm going to be presenting on work that was contributed by 2 EEPB grad students. So in the Johnson lab, we're really concerned with this main question of how do animal behaviors evolve? Because if we look across the animal kingdom, particularly in the breeding season in males, we see them perform these elaborate behaviors to either defend territories or compete for mates, or to simply attract and court mates like this foot-flagging frog or this acrobatic dancing golden collared mannequin. And in our lab, what we wonder is there must be some robust muscular traits that can provide not only the strength to perform some of these behaviors, but also the fine motor agility to perform these other behaviors. So we also know that these behaviors are highly correlated with testosterone. And we can see that if we look at testosterone levels across the breeding season. So we can see here with the mean testosterone concentration on the y-axis that in the breeding season across vertebrate groups, testosterone is elevated when we see these elevated social behaviors. We also know that testosterone affects muscles directly. It can influence muscle growth, muscle strength, and muscle performance, which means it's not super surprising if you look at the World Anti-Doping Agency, they list testosterone as an all-time banned substance for its athletes. So given the relationship that we know exists between testosterone and social behaviors in the wild and testosterone and its effects on muscles, what our lab is interested in understanding is that if testosterone mediates these behaviors we see in the wild through changing muscle morphology and muscle physiology. And this, the animals that we choose to study in our lab are the anole lizards because they perform these variety of social displays. So right now you're looking at 2 males, and the one on the bottom is extending his colorful throat fan called the dewlap. And right now he's head bobbing a little bit. And what you can't see is there's also females in these bushes that are also observing interaction between these 2 males. Now they're fighting. And usually there's squeaking happening, but I don't have the audio on. And then I don't know where they went. And then it looks at, so the male that was displaying earlier, he's saying to continue to display even though he's on this barbed wire fence. So that's kind of funny. So in our study, we specifically looked at the behavior and muscles underlying that dewlap display that male was showing on the previous slide, and also their copulatory behaviors. The muscle that controls the dewlap is the serrato-hyoid, which is this muscle that runs down the length of their throat. And when that muscle contracts, that flap of skin, which is attached to a piece of cartilage, bows out, revealing the throat fan. And then for copulation, if you didn't know, anoles have 2 penises called hemipenes. And so they'll actually, between mating events, they'll alternate which hemipene they use. And so there's one muscle that when it contracts, the hemipene ejects out of their follicle vent into the female. And then another muscle called the retractor penis magnus that when it contracts, it basically pulls the hemipene back into the tail. And so that's the muscle that we're going to focus on for this study. And so we specifically looked at these muscles and behaviors in 2 species of anoles, the brown anole, Anolis sagerii, and the green anole, Anolis carolinensis. And so what we did is we went out in Texas and we collected a bunch of males and females of both species, and we brought them back into the lab, and we assigned the males to one of 3 treatment groups. They were either given high testosterone, low testosterone, or they were a control. And so for that testosterone treatments, what we did, we removed the male's testes and inserted these capsules that could slowly release testosterone over a long period of time at a known concentration. And so after 6 weeks of this manipulation, we look at the male's behaviors in these arena trials, where we put a male in this arena, and then we put him with a novel female, and then separately a novel male to see how their behaviors change with testosterone. And as we expected, the ones with high testosterone behaved more than the ones with low testosterone. But the interesting thing that we wanted to look at was, okay, what's happening to the muscles underlying these display behaviors? So here I am showing one of the hemipenes, so there's 2 of them, for a brown animal. And I didn't label which ones had high testosterone and which ones had low testosterone. So I thought to be a little interactive, you guys could point with your right or left hand. Which one do you think came from a high testosterone brown animal? Okay, cool. I can't fool any of you. You're right. So the one on the right here is from a high testosterone brown animal. So it's kind of insane how over just a 6-week period, testosterone can have this massive of an impact on the copulatory organs. And then if we look at that retractor penis magnus muscle, we see that it's also increased in size. And the reason being is it's not that there's differences in the number of fibers between these 2 treatments. It's actually each individual fiber in the muscle is actually getting larger, which is pretty fascinating. So now to move on to the dewlap display in that serrato-hyoid muscle. So I'm just going to spoil it. We didn't see any differences in muscle size, which was kind of surprising because we see that they dewlap way more in the breeding season. So we kind of expected that there might be some change in the muscle size given the increased use of the muscle. But that's not the muscle size isn't the end of the story though. So my collaborators back at Trinity are continuing to work on this. So Grace is using these succinate dehydrogenase stains and these myosin stains. And what she can do is you can see that there's like different lightness and darkness to the pink stains. And what she can do is by comparing the 2 stains, she can identify whether each individual fiber in the muscle is either a tonic fiber, a slow oxidative fiber, a fast glycolytic fiber, or a fast oxidative glycolytic fiber. And so what might be happening is we might not be seeing changes in overall muscle size, but testosterone might be changing the composition of the muscles to account for change in use of the muscle. It's kind of similar to the idea of you wouldn't expect Usain Bolt to have a bunch of slow oxidative fibers. He's a sprinter. He's going to have like more fast glycolytic fibers versus a marathon runner who's expected to use these muscles for long durations and long periods. And then my other collaborator, Shauna, is currently looking at androgen receptor expression in the muscle fibers. So testosterone binds to androgen receptors. And what we know happens in that golden collared mannequin and foot flogging frog I showed at the beginning is that in the muscles controlling those displays, they actually have exaggeratedly elevated levels of androgen receptors, so that even with low levels of testosterone, those muscles are more sensitive to testosterone. So overall, what this study is able to do is look at testosterone and see how it affects muscle physiology at a variety of scales, from just the size of the muscle to the fiber composition, and then down to just receptor expression. And then we can take that relationship to start to understand how these animals are to be able to perform these behaviors in the wild. So with that, I want to acknowledge all of the other undergrads who were involved in collecting data for this project and give a big shout out to Michelle. She's an awesome advisor. And I will take any questions that y'all have.
Brilliant. That's a lovely talk. Thank you, Lauren. Do we have any questions from folks in the crowd? Well, Lauren, I have a question. Oh. Go for it. Well, how do you make those slides, Lauren? The actual histology slides? Is that pretty difficult to do?
Yeah, so the way I describe the histology slides, if you've ever been to a deli and you're looking at them slicing meat, so what we do is basically take these muscles and it's like like you basically have them at freezing temperature so that it's easier to cut them. And so you're cutting these thin slices, and each time you slice, it drops onto a slide. So we essentially get sections of the entire muscle, like lengthwise.
Oh, great talk. I was wondering, whenever you right? Oh wait. Sorry. I broke it. I was just wondering, whenever you had the slides with like the muscles next to each other, were they on the same scale?
Oh yeah. So let's go back to those slides real quick. They're not too far away. Yeah. So So they weren't so they're taking a different magnification, because the problem is the high testosterone ones are so massive that I had to take those at 4x. But the ones that were taken for the low testosterone were taken at 10x, but I scaled the images to account for those differences.
Thanks so much for the talk. I just have a question. How static are testosterone levels within individuals? Do individuals show any evidence of being able to ramp it up rapidly right before an immediate interaction?
That's a really good question. I don't know the answer to that, but what I can say is that, especially for these acrobatic mannequins, you do see this testosterone levels are elevated through the breeding season, but you see this like big spike like right before the breeding season starts, and then it kind of like it stays high, but it kind of decreases subsequently throughout the breeding season. So maybe that could explain something that's happening in animals.
Hi, we have a question from the YouTube chat. John Birmingham Jr. asked, do high testosterone lizards always win conflict?
Ooh, so that's a good question. So it has to do with, because oftentimes testosterone can make animals more aggressive, and whether that means you're actually going to win a conflict is iffy. So I'd say like not necessarily. That's a really great question. He's coming in.
Is testosterone expression mostly genetic, or do you know of any ecological or other factors that can go into testosterone production and expression?
I'd probably say, like, to my knowledge, I would say it's mostly genetic, but I'm sure there's like other things that can play into it. Because a lot of times, like when we manipulate these things like at a developmental level, it's usually like, oh, let's look at females, and if we give them testosterone early on. So whether there's like other environmental things, mostly it's us putting testosterone in everything, like keeping it out or moving it. Yes.
Brilliant. Let's get Lauren on the end.
No. No. Okay. Wait, have Tim and Tim camera on. Okay.
Okay, our next student to present today is Wen Guo. He did his master's and his bachelor's at Taiwan University. Is a fifth-year student in the lab of Ken Olson, and his talk today is titled "The Genetics of the White Clover Polymorphism in the White Mark Polymorphism in White Clover."
Thank you, Ari. So I'm Wenxi from Ken Olson's lab, and today I'm going to share a work. Hello. Hi. Okay, now it's on. So the work I'm going to share today is initiated from last summer with an excellent undergrad, Emil Cunningham. So it's about leaf mark polymorphism. And leaf mark is a kind of leaf variegation. And leaf variegation is the heterogeneous color pattern of a leaf. It is pretty common in the nature, and it's also popular in the house plants. And there are two kinds of leaf variegation. One is pigmentation. Oh, like here, they can be the structural color. So under this white color, they are like the intercellular space is bigger, and the patch of the mesophyll cell is loose than the others, so they can look white. And several adaptive roles have been proposed for these kind of variegations. but only a few of them has been validated experimentally. For example, like here, some people find this white mark can involve in thermoregulation. The white mark can increase the local leaf temperature in a cold environment. Or like here, it can involve in herbivore defense. Like this leaf, the white mark pattern mimic the leftover meal from a leaf mining moth larvae. And because the moths tend to lay egg on intact leaf, so if a leaf can pretend it has been eaten, it can avoid being the target of the leaf mining moths. And a very controversial effect is the Dazzle effect. So if there are white marks like a swaying in the wind, and it can create a dazzling and can confuse the insect or the herbivore approaching it. And here is an example like this. But we know this effect is hard to be proved. So like the function role is still like rarely being investigated. And also the genetic mechanism for this leaf variegation has been rarely touched. And an exception is the white clover, because the white clover has the leaf mark polymorphism. And in 60 years ago, like people did extensive crossing experiment between a clover with leaf mark and without a leaf mark. And the model they find is this is a single gene trait, a single locus with multiple allele, controlling the presence or the absence of the white mark. But even after the 60 years, the detailed molecular mechanism and the adaptive role is still elusive. So why is it difficult to study for the genetic part? It is because there is no reference genome for the white clover, so it's hard to study. And then the trait is not strictly binary. Between the individual, you can find there are some intermediate traits between, and even within a single plant, you can see a gradient of the leaf mark trait through the developmental stages. I personally find the leaf exposed to the sunlight is easier to develop the white mark trait. And for the adaptive part, for a typical adaptive trait, there is usually an association between the trait frequency of certain environment gradient, certain environmental factor, and we call climb here. And there's no clear climb found in white clover. And because of it, like the animal behavior experiment to test the functionality of this trait is hard to be conducted. So yeah, it's a trait not easy to study. And I'm very fortunate to have the opportunity to study this trait because I'm doing a drought tolerance experiment based on the white clover. So we constructed a pedigree mapping population from the two parents. So one parent from Gainesville, Florida, it has the strong white mark. And the other parents from Duluth, Minnesota, it is without any white mark. So the experimental design is to make crossing between the two parents. And then we get F1 and F2 and F3. And through the generation, the meiosis process shuffle the genetic material. And we can use tutorial mapping algorithm to find the association between genetic marker genotype and our trait, the leaf mark. And because of the linkage, the marker, the genetic marker should be close to the real causative gene we are looking for. And when the genetic material gets shuffled, I also find this binary tree gets segregated. So there are different degrees of leaf mark in the offspring population. Like here, this is a plant, an offspring with very strong leaf mark. And this is also strong. Then it gets weaker and weaker and can barely be seen. And still this one and this one. You can see there's still leaf mark here. So this makes the phenotyping process really challenging. And this one also very weak one. And this is the real phenotype with no leaf mark. And so here is the QTL mapping result. The x-axis of this plot is the chromosome number. The y-axis is the LOD score. Higher LOD score means there are stronger association between genotype and the phenotype. So we can see there is a super strong major effect QTL on the chromosome 5. And there are 6 other minor effect QTLs. If we look from each QTL, we can see they have the same effect. So the x-axis here is the genotype. The A means the allele from the GFL parent, and the B means the allele from BMR parent. And the y-axis is the label of the leaf mark. 0 means there's no leaf mark, and 4 is the strongest leaf mark. And the label between 1 to 3 is the phenotype in between. So we can see this is the marker, QTL marker, chromosome 5 is the major QTL, and the homozygote for A and AB is about the same. So it's almost a complete dominance effect for this major effect QTL. And it is pretty consistent. The allele from the GFL parents can be more on the leaf mark trait. And if we make the cumulative plot of the A allele, and we can see a very linear pattern of the trait when the sum of the A allele get more and more. And this is a very nice example of the quantitative trait. And if we look from the perspective of the major effect QTL, we can see there is a gradient, the minor effect QTL has the effect. It can make the weak mark pattern stronger, especially when the major effect locus is heterozygote. And we can also see there are some outlier here. And I think that's the limitation of our marker densities, because maybe there's a recombination between the genetic marker and the real QTL. But I don't think this noise is bad, because I think they can provide material for the fine mapping in the future. And after we find the QTL region, I'm moving forward and being ambitious, try to find the candidate genes. And with our limited data, with the whole genome resequencing data from 3 individuals, these GFL and the BMR, they are the parents of the pedigree mapping populations, and they are another exception from St. Louis. And the BMN and the STL, they don't have the leaf mark. So what I'm looking for is a loss of function like region in the genome, and they should be consistent among the BMN and STL. And then I find there is a 2 kb deletion on the upstream of an interesting genes. It has a cute name called Superman in rice and a rabbit ear in Arabidopsis. It is an organ identity gene in floral development. So it pretty makes sense, because if we reveal the white mark mechanism, it is a structure color. It is the arrangement of the mesophyll cell. So if the candidate gene is a developmental regulation genes, it is pretty likely. So to summarize, we find a major QTL on chromosome 5, and it could be a developmental regulation genes. And through the environment and other minor QTL, it can affect the penetrance, the strengths of the white mark trait in clover. So that's it. And I get many people to say, and this is the undergrad I'm working on. And I think because I still have a little time, I want to make a vote here. So do people think the white mark in clover, it could be an adaptive trait, or it is just a neutral trait? Like we should not dig too much into its trait. So the people who are adaptationists, Please show your hand. Wow, thank you. And how about the neutral? Think about the white mark just as schedule of the evolution history. Thank you. I think the neutral celery wins here.
Very divided room here. Man, that's a great talk.
So Wen, I loved your question at the end because it leads to the question I wanted to ask you. Are there any like negative effects to producing those white marks that you know, like why do you see some clover with them and some without? Like, why wouldn't they all just have it? If it could potentially be beneficial in the long run, like to avoid like larvae nesting in them.
Yeah, thank you. So like we were trying to find the negative effect of the leaf mark, but it's difficult to prove it. So at the beginning, we expect the leaf with white mark because white mark I intuitively think it's labeled chloroplast, so it may have lower photosynthesis rate compared to the leaf without white mark. But we try to measure the photosynthesis rate using a mapping population, so they have a homogenized genetic background. But the thing is like we only get higher variation in the leaf with strong white mark, and it's hard to tell they are different from the others. So yeah, I'm still thinking about a way to prove it. Yeah.
How large is that region, I don't know, metabasis and kilobasis on chromosome 5 for that large effect QTL? How big is that region?
So between the marker, between a major effect marker to the Jackson marker, it's about 300 kilobase pair, includes about like 30 genes. Yeah, so if I could just try out each gene one by one, maybe I can find it.
Nice talk, great work. You mentioned that there might be environmental factors involved in the trait. I'm curious if there's variation even within an individual, maybe if it's like sunlight hitting the leaf in a certain way. And then if there is variation, how did you pick a leaf to score? And then how did you score? Like, is it a visual thing or did you use some sort of imagery?
Yeah, it's not easy. So last summer, my undergrad and I tried to phenotype a same individual several times. So if there are no white mark all the time in very different trials, we say it's yellow, no white mark. And if it's a plant with weak leaf mark and with no leaf mark leaf, it's a combination between weak and no. We score it at 1. And if it is a plant with all leaf with weak leaf mark, we score it at 2. And if it is a plant with all strong leaf mark, we score at 4. And so the combination between strong leaf mark and weak leaf mark score at 3. But I agree there are some human bias in phenotyping process. Yeah.
Hi, I'm wondering if there are any geographic variations in white mark frequency in the region, and are they associated with any environmental factors like temperature or altitude or whatever?
Yeah, good question. So far, I haven't seen any literature about it. I hope one day I could travel and see if they are like a different frequency of this trait across a big geographic range. Yeah, I think if we can find that climb, that gradient, it's a good proof to be adaptive. Yeah.
We also have a question from YouTube from John Birmingham Jr. again. Have you considered looking for new markers in your chromosome 5 region that you could use to ID potential recombinants that could narrow your candidate region?
Yes. In the future, I hope to develop more dense marker in that region. And by the finite mapping, or maybe by doing a tiny region of the like GWAS like approach, like using association analysis, maybe we can narrow down to the genic label. But I think it's still a long way to go.
Great, let's take one more time.
Hello.
Okay.
Oh, sorry. Please introduce me.
Our third presenter today here is Brock Mashburn. Brock did his BS at Truman State University. Then he went and did a master's at Wassel. He is now a fourth-year student at WashU, and he is in the lab of Christy Edwards at Mobot. His talk today is titled "How Connected is the Globe? Biogeographic Surprises in Hibiscus." Brock, take it away.
Thank you. I forgot to put my title on my title slide. Yes, so my name's Brock. I'm in the lab with Christy Edwards at Missouri Botanical Garden. I'm also co-advised by Ken Wilson here at the Danforth campus. My interests are broadly plant conservation, speciation, and biogeography. For my PhD, I'm working on a group of species in the genus Hibiscus called sectional hibiscus. It's about 25 species, and they're interesting for a number of reasons. The first reason that they're interesting is they are the progenitors of the cultivars of Hibiscus rosacenensis that are common in the horticultural trade. These Hibiscus rosacenensis cultivars are hybrids of the species in the group that I work on, and you can see the really intense and beautiful color variation among the flowers here. And these cultivars are a multi-million-dollar business in the horticultural trade. My main interest in this group is because of their conservation concern. Of the about 25 species in the group, 3 are extinct in the wild, 13 are critically endangered, and 3 others are endangered. And then there are 5 species that are not assessed, and we will assess them. And I think that when they are assessed, they'll either be endangered or critically endangered. And so a large portion of my PhD work is conservation genetics focused on a handful of these species. But that's not what I want to talk about today. What I want to talk about today are some results that are hot off the press. And because I titled my presentation "Biogeographical Surprises," I felt like I had to stick with the biogeographical questions, even though my analyses were still running on Wednesday morning. And I thought they're not going to finish. I'm not going to be able to present, but they're done, and I'm showing them to you first. There's 2 sections that I want to talk about. One is the global biogeography of the group, and then I want to zoom in and look at some questions about local speciation processes within one clade of the group. So first, global biogeography. A really fascinating aspect of this group is their global distribution. We know that the group arose in Madagascar based off of work of collaborators, and there are 5 species that occur in Madagascar. There are 2 species that occur in East Africa, and then 4 species in the Mashburn Islands, just east of Madagascar in the Indian Ocean. Then we have 4 species in Fiji and the surrounding islands, and 8 species in Hawaii. And so this relatively small group, that's a pretty recent radiation, occurs really about as far as you can get across the globe. So I'm going to show you in a different way. First, we have the Madagascar and Mashburn species on the bottom left of the globe. And then the Fiji species occur here on the bottom right, and the Hawaiian species up on the top right. So it's about as far distributed as you can get on the globe. And what's even more interesting is that no species occur in this area of islands and continental, huge continental masses in between. So eventually we'll be asking questions about how this might occur, but not quite there yet. So first, I just want to talk about the biogeography first, the patterns. So if what we know of what we know of their distribution and their origin is true, which I think it is, I can formulate a basic hypothesis about how the species came to occur in the places that they occur. So they rose in Madagascar and maybe a distribution event to East Africa, and then of course to the Mascarenes, which are right nearby, maybe from the Mascarenes to Fiji, and then from Fiji to Hawaii. This seems to be the most parsimonious distribution. And so if we were to construct a phylogeny of this group, we would expect the Hawaiian species to be the youngest, the most recent, and nested within maybe the Fiji species, which are then nested within the Mascarene species. And sister to all of those should be Madagascar species. So on the next slide, I'm going to show the results for our global phylogeny that just came out. And we rooted the tree with this Madagascar species that we know is basal to the group based on the river. And what we see here is that there is a monophyletic Hawaiian clade. But what's interesting is that this monophyletic Hawaiian clade is sister to the remaining species. We have more Madagascar and East Africa species next to it. And then nested within that is the Fiji species. And then nested within the Fiji species is the Mascarene species. So essentially what this biogeographical distribution or what this phylogeny seems to be showing is that we know that the group arose in Madagascar. And then there was an independent dispersal event to Hawaii. More speciation within Madagascar. Distribution dispersal to East Africa. Then an independent dispersal event to Fiji, skipping over the Mascarenes. And then from Fiji back to the Mascarenes, next door to Madagascar, more recently, I presume. So this is absolutely a surprise. And what's even more interesting is that previous studies that are a little bit older now and not quite accurate, at least propose a hypothesis that this group was only 1 million years old, which I don't think is accurate, but anyways. So another outcome of this phylogeny that I already sort of mentioned is that the Hawaiian group and the Mascarene groups are monophyletic. So we can use these groups to test speciation hypotheses within archipelagos, the archipelagos on which they occur. And so for the Hawaiian archipelago, I'm working with a really fantastic undergrad named Sophie Dorison. And it's turning out to be a kind of difficult and complicated. So I'm not going to talk about that. I'm going to talk about the Mascarene group. And this is the analysis also that was just finishing yesterday. And so I'm showing it to you here first. So local speciation processes within the Mascarenes. The Mascarene Islands are three main islands east of Madagascar. Rodrigues is the oldest island. It's about 15 million years old. Mauritius is the next oldest, about 8 million years old. And then Reunion is the youngest. It's estimated to be about 2 million years old. And it's also the largest. And actually, there's still an active volcano on Reunion. And I was able to travel to Rodrigues and Mauritius and collect in 2018. And then COVID. And then I actually got to go to Mauritius and Reunion in April of this year and do finish my collecting there. And I got to see the lava flow as well. So we have 4 species of hibiscus sectional hibiscus in the Mascarenes. Rodrigues has 1 species, Lilea forest. Mauritius has 3 species, Boreanus, Fragilis, and Genevii. And then Reunion has 2 species, Boreanus and Fragilis. Now this is the current understanding. I think it's going to change, but based on this understanding, you'll see that Mauritius and Reunion share 2 species. And so a sort of basic hypothesis about how these species came to exist might follow what we call the progression rule. And the progression rule basically states that there should be concordance between the age of the species and the age of the islands that they occur on. And so what we might expect and what the global phylogeny seems to show is that the species that arrived here probably came from Fiji. And we might get some speciation on the oldest island before Mauritius or Reunion even exists. And then dispersal to Mauritius as it sort of comes into existence. Speciation there, and then dispersal to Reunion. So that's sort of a basic hypothesis. Because two species are shared between the islands, we might expect maybe two independent dispersal events from Mauritius to Reunion. So what does my recent phylogeny show? Our basal species is Hibiscus liquoris from Rodrigues, the oldest island. That's nice. The next species is Hibiscus genevii, which occurs on Mauritius. And that seems to indicate a dispersal event from the oldest island to the second oldest island. Our next species is Hibiscus fragilis. And even though it's not shown as monophyletic here, at least currently it's one species, and we're gonna have to do stuff to it, but depending on how things shake out. And that's also on Mauritius, so that seems to indicate speciation on the island of Mauritius. Next, we have a monophyletic group called Hibiscus boreanus from Mauritius, so indicates potentially more speciation on the island. And then our next two groups, we have a red entity right now called Hibiscus boreanus, and that is on Reunion, and it indicates dispersal event from Mauritius to Reunion, the youngest island. And then we have another monophyletic entity here, still called Hibiscus boreanus in Reunion, and that indicates speciation on the island of Reunion. So more or less, what we seem to have is something that follows the progression rule, what we would have predicted. And here I'm just showing a very quick structural result to show that mostly the species seem to be pretty good, but there seems to be some like gene flow or hybridization going on. But I don't know what's going on. Maybe I feel I just saw this for the first time yesterday. So okay, so the last thing I want to say here too is that this seems to be a really great example of a remarkable plant radiation on an archipelago with dramatic shifts in flower color. And it could be the foundation for future studies about the genetic basis of flower color shifts and maybe like ecosystem or pollinator shifts or something like that. We'll see. And so with that, I'll just finish with some photos of pollinators or nectar robbers of Hibiscus boreanus on Reunion. Thank you.
Great talk, Brock. Thanks so much. I actually have a question for you. It seems like more and more we find that when integration is present, we completely misestimate the species tree.
Yeah.
And when you're trying to do biogeography,
Yes.
pretty good idea on
Yeah.
solid species tree. So
Yeah.
what gives you confidence or what doesn't give you confidence in your species tree? And then what are you going to do to resolve some of those outstanding issues if so?
What gives me confidence? So not much yet, I'll say that. I mean, okay, so the structure results give me some confidence for most of these entities, but you can see that in this area here, something weird is going on, and that is possibly leading to the fact that this thing, which morphologically is quite united, but we have a population in the south of the island, a population in the north of the island, which is all that's left of the species. And then these two populations exist like a few miles away from each other. So there seems to be potential for gene flow there causing problems in the tree. But how am I going to resolve that? I don't know yet.
I'll accept it for now.
Okay.
Great talk, Brock. I have a question, back where you're showing kind of the progression effect with the phylogeny. So it's, it looked like you saw
That's-
evidence for the, Boreanus, but like, what about, weren't there two species that you said you thought might have dispersed? to
Yeah,
that?
sorry. I realized that as I was saying, it's a complicated answer. It's a great question. Let's see. So this hibiscus fragilis was reported to exist on Mauritius, and I went into the field and proved that it doesn't exist, basically. So yeah, that's like a whole talk of its own that I don't really want to go into. But, in my tree here, I showed it as two beforehand, but people were saying that like one of the populations within this was hibiscus fragilis. And I should have been more clear on that, but good question.
We have to move on to the next talk, but let's take Brock one more time. Our last student speaker of the day is Anna Wassel. Anna did her undergrad at Emory University. She is a third-year student in the lab of Jonathan Myers, and her talk today is titled, "Why do pawpaws do the things they do? Foundation species effect on community assembly." Anna.
All right, thanks, Ari. Yeah, I'm presenting the data that I collected 2 summers ago, so just over a summer, that basically aims to get at the why behind the what do dominant species do to community assembly. And I know we're getting towards the end of the day and y'all are ready for food, but this does feature our very special guest, the pawpaw tree. So it's an edible fruit, if that makes you feel better. All right, so first up, I want to get on the same page about what exactly I mean by a dominant species, because despite being used a bunch historically throughout literature, people rarely define it. So for today, dominant species are those that have a large relative population size in their community and a large impact on their ecosystem. It is possible to have a large population size and not a large impact, and it's also possible, as is the case with keystone species, you can have a large impact on the system, but a relatively small population size. So, sorry, keystone species, you're not dominant species today. By community assembly, I essentially mean which species live where and with whom and why. So there's a lot going on in this figure, but it mostly shows which species live where and with whom, and the why are the processes that are shown here. So we have dispersal and chance, also known as ecological drift, and multiple forms of selection going on. So those are our big 3 processes shown in this figure. If we're going to talk about the why dominant species do the things they do, we got to first cover the what do they do. In a meta-analysis of removal experiments of dominant species, Avolio and colleagues showed that when you get rid of dominant species, ecosystem function decreases. So that's going to be things like nutrient cycling, nutrient storage, et cetera, et cetera. They also looked at community responses. So those are things like species richness and evenness. And this was a little more uncertain. Sometimes it had a positive effect or a positive response ratio. Sometimes it was neutral to slightly negative. But when you took the absolute value, then it was significant. So basically that says whether or not it's a positive or negative effect, it is important. In short, the what is contingent. So maybe we'll have more luck with discovering consistently the why do dominant species do the things they do. In other words, we're going to be looking at the major processes of how dominant species affect selection and drift. And to do this, we came up with some fun process-based hypotheses. The first is our selective sweep hypothesis, which basically states dominant species will increase the relative importance of ecological selection. as opposed to increasing the relative importance of ecological drift, as shown in our second. And for each of these, we have some very specific predictions. First up, for local diversity, also called alpha diversity, we're expecting lower alpha diversity in areas with dominant species, and also pretty low beta diversity. By beta diversity, I mean species composition change. And that'll be because the species composition will be less random. Certain species will be favored due to their competitive ability. Meanwhile, for drift, we would also expect lower alpha diversity, but we would expect higher beta diversity, so more compositional changes, because species will be a more random draw from the species pool. So we tested this with our very special guest, our dominant species, the pawpaw tree, Asimina triloba. You can see the edible fruit up there in the left. If you haven't had it, it's very tasty, I recommend. Its geographic range is all over the eastern half of the United States, but at a local level, as seen here at Tyson Research Center, this is about a 20-meter scale in between these two black dots. They form these really dense, very discrete clonal colonies. So that makes them awesome for me. It makes it very easy to study the presence and absence of dominant species. So to test these hypotheses, we had some fun in the field at Tyson Research Center, more specifically our forest dynamics plot. And we chose 5 blocks. Each block had a pawpaw patch and an area without pawpaw that was abiotically similar. And in each of these areas, we sampled 5 1-meter quadrats. So we got the identity and abundance of all understory vascular plants. So all the little cute herby stuff. So that gave us a total of 50 quadrats to assess the alpha diversity within each plot, and also the beta diversity among plots within each biotic type. So what did we find? For alpha diversity, we found 79 total species. 73 were found outside pawpaw patches, but only 38 were found within the pawpaw patches. Thankfully, this pattern was confirmed by statistics. Even when we controlled for block level effects in a mixed effects model, it was significant. We got that handsome p-value to prove it. This fits into our hypotheses. Both are supported. So that's good, but we're going to have to dig a little deeper to really understand what's going on. So this is where it gets a little more complicated, but also a little more interesting. Here we have the beta diversity between the red, which is outside pawpaws, and the blue within pawpaws. They're not statistically different. In math words, that means that the Bray-Curtis distance to centroid is statistically non-significant. In human words, it means the composition from one control plot to another control plot, the differences are just the same as if you were to go from one pawpaw plot to one pawpaw plot. Great. We then built a null model that basically shuffled up all our species, placed them in random in different plots, and that simulated random assembly. So if it were just completely drift, what would the communities look like? For pawpaws, it wasn't significantly different. But for our outside plots, it was different. Here we see the effect size saying that, okay, maybe pawpaw is a little above what we would expect from total neutral assembly, which is this dotted line. But those outside plots they're really up there. They're even more different from plot to plot than we would expect by random chance. So our second prediction wasn't supported either way, but our third one seems to support this ecological drift hypothesis because species were more random in pawpaw plots compared to outside. And that makes sense because the community size within pawpaw plots was significantly lower. We're talking on average, what is that? Maybe like 15, 20, versus outside, it was close to 50. And thanks to math and Hubble and Veland and other great scientists, We know that when you have a lower community size, the effects of drift are more likely to be apparent. You have more random extinctions. So my parting words and pictures, my conclusions is that this study seems to suggest that locally dominant species increase the role of ecological drift at local scales. So up next, I want to study more spatially explicit questions. So things like, how does the size of the patch affect things? Are there maybe edge effects going on? I also want to include the role of dispersal, because that was the last big process I didn't include. And I'd also love to analyze some functional diversity. I want to thank all the people and places and animals that helped me both logistically and emotionally through this process. And I would love to take any questions.
Great talk, and can you just humor me?
Yeah.
As a herpetologist?
Yeah.
Can you speak a little bit about the biology and the psychology of pawpaws and why you think they are such a dominant species? Like, what are they
Yeah.
actually doing?
Absolutely. We can go back to our pawpaw slide. There we go. So one thing is they are the northernmost species in an otherwise tropical family. So you can kind of tell from this canopy photo here, they have these massive tropical leaves that really create an unusually shady environment. We know that anecdotally, I'm starting to get canopy photos to test that mathematically. And they also seem to only grow in valleys with really wet soils. So essentially, they create conditions that just keep community sizes low, and that's why drift is the way it is.
Thank you.
So going back to the beginning when you were saying like all the species that you found, and I think there's like 79, but only I think 73 or so were found outside. So I assume that those 6 species difference were only found in the pawpaws. Do you think that there's some disadvantage that they have and in these like outside pawpaws that might have like increased selective pressure against them or something like that?
Yeah, potentially. One of those 6 species is invasive. It was Euonymus fortunei, I believe. So an invasive vine that I think was only found maybe once or twice throughout the whole study. And the other ones were pretty poorly defined morpho species or just like really damaged and hard to identify. Yeah, so there is a chance that there is a unique environment in the pawpaw patch that would give refuge to maybe 1 or 2 species, but it seems to keep population and community sizes so low that it seems unlikely.
I kind of have a question about like following up on the natural history side of things. So what are the phenology of the pawpaw trees and then the herbs that pop down with them? And do you like being there first or being there later influence like how the community is influenced?
Oh yeah, great question. So I do know that pawpaws, despite being tropical, drop their leaves a little later than you would expect and leaf out pretty early. And I would expect that would have an effect on the herbaceous communities, but I personally haven't looked at that yet. But yeah, they do flower in the spring and fruit late in the fall and are eaten by a lot of animals and get dispersed. So you would think maybe those dispersal animals would disperse other species, but as of right now, it doesn't look like that. So.
Hi, Anna.
Hi.
I think it was your alpha diversity results. You showed the data for each of the 5 blocks. And I know you accounted for them in your random effects model, but it looked like blocks 1 and 5 had like a much more dramatic difference, and the middle 3 blocks it was more subtle. And so I'm wondering, were there some spatial effects going on? I mean, were there notable spatial differences? Are these blocks all in a line? Are there like some edge effects or some differences in abiotic properties among the blocks that might explain these? Like, what's going on?
Yeah, great question. So we tried our best to control for abiotic conditions by running a principal component analysis on the data that we have for this whole swath of forest. So the general like slope, aspect, soil should be pretty similar.
So yeah, after that, I don't really know. It might be really small scale abiotic differences, which is what I suspect and what we're starting to collect data on to test going forward.
Okay, let's thank Anna. So this concludes our student seminars. Thank you to Usam and Matt for helping facilitate the IC seminars, and we'll pick back up to next week. And reception, don't forget about that, and there afterwards.
Putting up.